Welcome everybody, my name is Doug Rains. I am the Program Director for Genetic Testing at Merit Laboratory Partners in Franklin, Tennessee. And what I want to do in this video series is I want to go over some basics of pharmacogenetic testing. This video and the next video will focus on just providing a basic intro to genetics. So I'd like to start with the question, what is DNA? Well, this is just a very long series of molecules consisting of a sequence of 4 bases, those bases being the molecules. We call them by their abbreviations, G, A, T, and C. And if you look down here, this is just a linear molecule of DNA. It's actually a pretty short one, and you can see that it consists of these different G's, A's, T's, and C's in a certain order. That order may look random. Trust me, it's not random at all. Now, these bases, as we call them, they're actually 3 billion per human genome, and they're divided among 23 chromosomes. And I should mention that each of your nucleated cells contains a full set of these chromosomes. Pretty amazing. The chromosomes themselves are then divided into genes, which are unevenly spaced along the length of those chromosomes. And there's something like 23,000 genes in the human genome. I want to touch on something that's known as the central dogma of genetics. And I'm going to give you the very simplified version, but the relationship I'm going to show you here is extremely important. And that relationship is this. DNA leads to protein. Or, to put it another way, protein is derived from DNA. Now, more specifically, DNA sequence or gene sequence leads to protein sequence. So if the sequence of DNA changes, it's possible that the sequence of the protein will also change, along with that protein's activity. Well, just a couple of quick terms relating to what we just discussed. DNA sequence, which again has a major impact on protein sequence, we refer to that as genotype. So we will talk about, say, a patient's genotype for a specific gene. That's just the sequence. Whereas the resulting trait is the phenotype. You might think of phenotype as the outward manifestation of genotype. Now that's not entirely true, only because proteins aren't the only influences on phenotypes. You also can consider things like diet of a person, their personal habits, for example, whether or not they smoke, plus environmental influences. But all of these combine together to form phenotypes. Phenotypes come in many different forms, and some of these can be visually apparent, such as eye, hair color, height, that sort of thing, or traits can be completely invisible to the naked eye. For example, how efficiently a particular enzyme functions, and that's going to be one that we're particularly interested in with pharmacogenetic testing. But that raises a really important question. How do we measure traits that we can't actually see? Well, this is where genetic testing is such a powerful tool. Just by determining a person's genotype for certain genes, we can often make very accurate predictions about the resulting phenotypes. In fact, that's the basis of pharmacogenetic testing. Alright, well that's it for this first intro video, and we'll continue with this discussion in part 2. Hope you'll join me.